the soil? What does it do to the soil ecology?
The GMO glyphosate combination, the GMO translocates the glyphosate throughout the plant. It's released then through the root exudates so that the glyphosate then is actually contaminating or introduced into the entire root zone of the plant. That may be just a foot or two, or it may be 25 or 30 feet, as you see with alfalfa. Even wheat in the Pacific Northwest has a root system 25 feet deep. So you're spreading that chemical, powerful antibiotic, powerful chelator, well beyond any area of the soil where there's an opportunity for microbial degradation.
What happens to the microbes in the soil when the glyphosate comes?
Glyphosate is a very powerful antibiotic that's toxic to the beneficial organisms that are responsible for soil aggregation and for soil health, but it stimulates the pathogens.
Talk about what happens when the antibiotics get to the microbes. What do they do?
Well, antibiotic glyphosate, when it gets to the microbes, acts just as any other antibiotic that they would be sensitive to, and it totals them. It kills those microorganisms or weakens them, changes their activity, their ability to survive compared with other organisms that are also in competition with them.
What happens when those microbes die? What happens to the soil?
When you take that group of organisms out of the soil that are responsible for soil aggregation, we lose our soil structure that's so critical for air movement, for nitrogen movement, and for water percolation into that soil, so that we essentially move.
Impacts on the soil. What does it do to the soil ecology?
The GMO glyphosate combination, the GMO translocates the glyphosate throughout the plant. It's released then through the root exudates so that the glyphosate then is actually contaminating or introduced into the entire root zone of the plant. That may be just a foot or two, or it may be 25 or 30 feet, as you see with alfalfa. Even wheat in the Pacific Northwest has a root system 25 feet deep. So you're spreading that chemical, powerful antibiotic, powerful chelator, well beyond any area of the soil where there's an opportunity for microbial degradation.
What happens to the microbes in the soil when the glyphosate comes?
Glyphosate is a very powerful antibiotic that's toxic to the beneficial organisms that are responsible for soil aggregation and for soil health, but it stimulates the pathogens.
Talk about what happens when the antibiotics get to the microbes. What do they do?
Well, antibiotic glyphosate, when it gets to the microbes, acts just as any other antibiotic that they would be sensitive to, and it totals them. It kills those microorganisms or weakens them, changes their activity, their ability to survive compared with other organisms that are also in competition with them.
What happens when those microbes die? What happens to the soil?
When you take that group of organisms out of the soil that are responsible for soil aggregation, we lose our soil structure that's so critical for air movement, for nitrogen movement, and for water percolation into that soil, so that we essentially move.
Impacts on the soil. What does it do to the soil ecology?
The GMO glyphosate combination, the GMO translocates the glyphosate throughout the plant. It's released then through the root exudates so that the glyphosate then is actually contaminating or introduced into the entire root zone of the plant. That may be just a foot or two, or it may be 25 or 30 feet, as you see with alfalfa. Even wheat in the Pacific Northwest has a root system 25 feet deep. So you're spreading that chemical, powerful antibiotic, powerful chelator, well beyond any area of the soil where there's an opportunity for microbial degradation.
What happens to the microbes in the soil when the glyphosate comes?
Glyphosate is a very powerful antibiotic that's toxic to the beneficial organisms that are responsible for soil aggregation and for soil health, but it stimulates the pathogens.
Talk about what happens when the antibiotics get to the microbes. What do they do?
Well, antibiotic glyphosate, when it gets to the microbes, acts just as any other antibiotic that they would be sensitive to, and it totals them. It kills those microorganisms or weakens them, changes their activity, their ability to survive compared with other organisms that are also in competition with them.
What happens when those microbes die? What happens to the soil?
When you take that group of organisms out of the soil that are responsible for soil aggregation, we lose our soil structure that's so critical for air movement, for nitrogen movement, and for water percolation into that soil, so that we essentially move. Well, our concept of genetic engineering, as we understand it now, is quite drastically different from what we had 50 years ago. 50 years ago, we thought that you had one gene and one function. What we know now with genetic engineering, what we're doing is we're splicing in additional genetic material so we disrupt the integrity of the genetic code. When we splice in that genetic material with our current concept of genetics, we change the spatial relationship between the adjacent genetic material, and then as it's influenced by the environment, we have many different products that are produced from that one piece of genetic material that's gone in. But if it's Liberty Link or Roundup Ready or any of the other herbicide tolerant traits or the Bt insecticide production traits that we're inserting those genes for, they go in at different locations because we don't have the precision to know exactly where they're going. So it changes the spatial relationship between those genes, and therefore they produce different products. They may produce the product that we want, tolerance to glyphosate or Roundup, or tolerance to Liberty, which would be glufosinate, but we have no way of predicting what other products might be also produced. So just for the Roundup Ready trait, we know that there are up to 60 other proteins that are produced in addition to the enzyme that gives that plant its ability to tolerate glyphosate, a very broad spectrum herbicide, so that those other proteins then have some probability of toxicity that we've never studied, because most of those are completely unique proteins. They're new products. We've never seen them before in nature. That was a serious problem for us with genetic engineering L-tryptophan, for instance, as a nutrient substance. Actually, a company in Japan was manufacturing it as a nutrient substance for the United States, And in genetic engineering their bacteria, even though they were using genes that were from the bacteria itself, and they were merely duplicating them and increasing the ability of that bacteria to produce the L-tryptophan supplement, it was also producing another product that turned out to be very fatal and lethal to people who consumed that product. So
excuse me, so is it accurate to say then, is that correct?
You know, when the gene that gives the plant tolerance to glyphosate is inserted, then that plant becomes resistant to the glyphosate. What they're doing is inserting a whole nother physiologic pathway that isn't shut down by the glyphosate. So you can spray then a genetically engineered or a Roundup Ready plant with glyphosate without killing it, but you kill all the other plants that are around it, because glyphosate is a very broad spectrum herbicide, weed killer, that essentially kills every plant unless it's resistant to it. So, glyphosate is an EPA approved broad spectrum herbicide, weed killer. Essentially kills all weeds except those that have been engineered to tolerate it, or now we have some resistant weeds that have developed their own means of resistance to overcome the toxicity of the glyphosate. Glyphosate, or Roundup, is a very unique chemical. We consider it a 100-year chemical. It'll be 100 years before another chemical with this broad spectrum activity will be invented. It's literally a very unique compound, very simple compound, but has very unique characteristics in many areas. It was first patented to clean steam pipes and boilers as a very powerful chelator. 10 years later, it was patented by Monsanto as a very broad-spectrum herbicide, weed killer. In 2000, it was patented as a broad-spectrum antibiotic, so that it has many activities, but it's a unique compound. Now, when Monsanto patented as an herbicide, as a weed killer, they had one study which said it acts on the EPSPS enzyme, which only plants and fungi and microorganisms have.
And what does that mean? Back up just a second.
That enzyme is for the shikimate pathway, which is a pathway that humans and animals don't have. So an EPA asked about the safety of this compound that Monsanto asked to have registered as a broad-spectrum herbicide initially in 1974. Monsanto said, well, it only inhibits the EPSPS enzyme, which humans and animals don't have, so therefore it can never be toxic to humans and animals because it doesn't shut down any of their essential functions. Therefore, they said there's no need to do any toxicology studies on it because it can never be toxic because it is so specific for this one enzyme.
So then what happened?
Now, we know since that time that actually this broad spectrum chelator, mineral immobilizer, in other words, a chelator, first, when glyphosate was first patented, it was patented to see as a very strong chelator. In other words, a chemical that can grab on to other minerals without becoming a compound with that mineral, but it can change the characteristics of the mineral. So when it grabs onto it, it can increase its solubility or it can reduce its solubility and make other changes. It'd be kind of like having a parking place that is an adequate size for a sedan or a car, and then all of a sudden when you chelate that car and you double the size of it with this other compound that grabs onto it, you're having to try and park a semi in a spot that would normally be adequate for a car. So that a chelator is merely a chemical that can grab onto another one and change its characteristics. That's why when it grabs onto the calcium or magnesium in your shower or in a boiler, it can descale it because it can grab on and move it off the walls. As a weed killer, it does the same thing. It grabs onto an essential mineral and it immobilizes it. In other words, it makes it so that that mineral, which serves as a key, if you look at the example of having a big powerful engine in your car or a tractor or a truck, can do a lot of work for you, but it doesn't do any work until you turn the key on. So that what happens then when you have a strong chelator for that mineral that is the cofactor or the key for that enzyme is that all of a sudden it doesn't fit in the keyway. So that you have essentially pulled the key out of the ignition and you shut down that motor that we refer to as an enzyme.
And what does that mean? What does that
So that shuts down the physiologic pathway then. That motor isn't going to be able to do any work. And so everything starts shutting down from that point on in the physiological pathway, in the stream of events that take place in biology. You've essentially stopped it.
And what does that? How does that affect it when you eat?
Okay. Now, what that does is, if you were relying on that food, for instance, that's been treated with glyphosate, if you're relying on it as a source for your minerals, those minerals are chelated and they're not available then for your body to use for your enzyme function. So you become deficient in those particular minerals.
So then glyphosate
Glyphosate shuts down.
Yeah. By chelating the mineral, glyphosate shuts down that engine that provides function for life for the plant.
Okay. And so
Go ahead.
Okay. And as a chelator then, glyphosate can grab onto the minerals. Now it grabs onto the positively charged minerals. That would be iron, manganese, calcium, magnesium, cobalt, nickel, zinc, copper, any of those positively charged elements that are very essential for our functioning or for the plant's functioning or bacterial functioning. And all of those minerals then can be immobilized or the keys pulled out of the ignition for all of those enzymes that those minerals serve as the key for. That means that over 291 enzyme systems are shut down by glyphosate. It's not just the one. It's not just the EPSPS that they initially told EPA was the only one that it's active on. It's active on any of the positively charged mineral elements that are essential for life.
Okay. I have to turn it
If you
on.
remember that slide that I have, where I show the motor, the engine over here, and the key. The motor is the enzyme would be as the motor. The key, very small entity compared to this big engine sitting over here, but the key is the cofactor or the catalyst that turns that engine on. So copper and manganese and magnesium, all of those minerals then serve as the regulatory activity for all of these engines. They're the key that turn them on or turn them off. When that mineral is no longer available or is immobilized, or if you're just deficient in it because it's not available in your food, then those engines can't run because they have to have the key turned on. So when you add a strong chelator like glyphosate, you essentially grab onto all of those minerals and you pull the keys out of the ignition so that those motors then shut down.
And these are all minerals that conduct electricity, right?
Well, they could,
correct?
some of them could conduct electricity. They're metallic, many of them metallic, but calcium doesn't conduct much electricity. Magnesium, I mean, you can get a current through a ionic flow, but glyphosate can grab onto magnesium just like it can manganese or copper or zinc and immobilize them or pull the key out of the engine so that all of those functions then stop.
Okay. You got it.
When Monsanto went to register glyphosate, they stated that it was active only on one enzyme, and that was an enzyme that humans and animals don't have. So they said it has to be safe. It can't be toxic because it only works on this one enzyme. I mean, if you want a comparison of short-term versus long-term, the tobacco toxicity would be a comparison here. It'd be like taking a person that was using tobacco product for a week or maybe 3 months and saying, well, there's no real effect here. The body's pretty tolerant and we don't see any accumulative damage. So, but then you take that same individual and follow the usage for 10 years or 20 years, and you see the chronic effect then, and that accumulative effect. And that would be typical that we would see with almost all of our carcinogens. A carcinogen, very cancer-causing chemical, rarely causes cancer on a short-term basis. It's a chronic exposure, chronic involvement that leads to the development of cancer.
So the same is true of glyphosate then?
So, and the same thing with glyphosate that you see. As an herbicide, glyphosate has been a powerful tool for agriculture because it's systemic in the plant. You can spray it on one part and then it distributes itself throughout the plant. It accumulates in the shoot tip, in other words, in the actively growing parts of the plant, so that it accumulates in the seed that we eat, it accumulates in the new fresh growth that we would eat or our animals would eat. But about 20% of it then will move down into the root system and out into the soil. The way that it kills the plant is to shut down the defense mechanism of the plant so that the plant then becomes very susceptible to soil-borne pathogens. What it's essentially doing is it gives the plant a bad case of AIDS. The plant can then no longer defend itself against all this myriad of organisms that are readily available and anxious for a place at the dinner table. As it moves out into the soil, it kills off the beneficial organisms that do have a shikimate pathway. Those are the organisms that normally suppress the disease organisms. They keep them in check for us in nature, so that it kills those organisms off and it stimulates the disease organisms. So that what we see overall is that the plant is deprived of minerals as the glyphosate moves down into the root system, so that those keys for its defense mechanism are being pulled out, so that the plant becomes very susceptible to disease. It stimulates the disease organisms become more active, and it also deprives the plant of replenishing those minerals to restore its resistance at the same time that it's toxic to those organisms that would help the plant defend itself. I became interested in glyphosate as an herbicide because we know that all of our herbicides are mineral chelators. They have an effect on plant nutrition. For 55 years, my research has been dealing with natural biological controls for many of these soil-borne diseases, and nutritional control because we don't have good genetic resistance or chemicals that we can control many of them. So when we look at a new herbicide, we look at those diseases that might be stimulated. So an example would be with the phenoxyprop herbicides or tordon. Those are copper chelating elements. In other words, they reduce the availability of copper. In the Pacific Northwest and the Canadian prairies with wheat and barley, we have a disease that can attack the head of the flower of the wheat kernel and essentially destroy it. It produces a fungal sclerotia then that you have a toxin in that grain that is produced. And normally the plant is resistant because wheat and barley are both self-pollinated crops, and the glooms will prevent this fungus from the spores from being able to penetrate into the pistil. If the plant goes has a copper deficiency though, then the wheat wants to do all that it can to reproduce, so that if it's copper deficient, it becomes sterile on the male side. The pollen becomes sterile so that it can't fertilize the pistil. So the plant then, in trying to fulfill its measure of creation, opens the floret so that it can receive pollen from outside. When that happens, it also loses its resistance because then the spores of the fungus can readily blow in and infect the pistil. So with copper deficiency, if you have an increase in the disease called ergot, that's this fungus disease, we know that it's copper deficient. And so if a farmer is going to use the phenoxyprop herbicides or tordon, any of that whole group that are copper chelators, we tell them make sure you compensate for that damage. So, and if they do that, they can use the herbicide effectively for weed control without having the unintended consequences of increased disease and lower yield. So when glyphosate was approved in 1974, I had some research going on with different herbicides and looking at those because this was a new class of chemical, new class of herbicide that we didn't have much information on. And the first thing that we started seeing was an increase in a disease that we refer to as take-all. That's caused by a soil-borne fungus, and it's primarily, it's the resistance to this particular soil-borne fungus depends on an availability of manganese. So that as soon as we started seeing an increase in this disease where glyphosate had been applied the previous year, even though it wasn't used for wheat or barley, if it had been applied the previous year as a burn down, then we saw an increase in this disease. So I became interested if this was a specific herbicide that just chelated manganese. Didn't take very long to realize that it was chelating many minerals because it chelates all of the positively charged minerals. But manganese is one of the very important ones as far as the take-all disease. So in 1975, I started setting up research plots then so that I could monitor the nutrient status of plants. And then in 1996, with the genetically engineered plants, I recognized, or I hypothesized, that if we're going to be putting this very broad spectrum chelator that's going to grab onto a lot of different nutrients directly onto the plant, that it's going to have a lot of genetic inserts in order to offset the effect of this broad spectrum chelating effect. So that we saw then that glyphosate was reducing the mineral availability for all of these cations. We saw that it was increasing the susceptibility, not just to take-all that involved the manganese, but we see over 40 diseases that are more severe where glyphosate has been used in the weed control program even 2 and 3 years before the particular crop that we're looking at that disease on.
Okay.
So, and what you need to recognize in the genetically engineered plant is that there's nothing in that plant that does anything to the glyphosate that you're applying. Glyphosate is still a very broad spectrum chelator to immobilize many nutrients. The only thing that the genetic engineering does is make it possible to apply the chemical directly to the plant without killing the plant. But it doesn't change the chelating activity of the glyphosate, so that the glyphosate is still reducing the availability of nutrients in the plant. In fact, we'll find within 6 hours in sugarcane, where it's used as a ripening agent in some countries, that within 6 hours, the manganese available in the sap is reduced by 90% because it's chelated by the glyphosate. You'll see that with the other nutrients. So there are many other effects of the glyphosate when it's applied to the plant, but one of those major effects is to reduce nutrient availability then from a food or a feed standpoint, as well as nutritional standpoint for the plant itself. The most insane use of it is as a desiccant, as a ripening aid, because you're applying glyphosate directly to a food product at a late stage of development, so that the glyphosate only has one place that it can go, and that place that it accumulates is in the seed that we're harvesting then for food and feed. So the concentrations of glyphosate that you're going to be taking up are going to be quite intense compared to what it would be if it was just used for regular weed control. Glyphosate is a very unique chemical. It's very simple, fairly cheap to manufacture, but a very difficult chemical to degrade. It's detoxified in the soil because it grabs onto calcium and magnesium so that its herbicidal activity is reduced, but it's immobilized those nutrients at the same time. So it can impact our overall food production and the food nutrient density system in a number of different ways. It's a very strong antibiotic, so that it's toxic to the organisms in the soil that make nutrients available for plant uptake. It reduces the efficiency of nutrient uptake in the plant because it's a chelator. It can accumulate in the soil for 22 to 30 years and then be desorbed, but as it's accumulating, it's also immobilizing other nutrients so that it's reducing their availability for the plants to take those up. Doesn't have to be genetically engineered or not. Those nutrients aren't as readily available for any crop that's grown on that soil as long as the glyphosate is present. A half-life may be 1 to 2 years, or again, it may be 22 years. So we're talking, in some cases, even generations, that the glyphosate can still be present and have its effect on subsequent crop growth.
Okay.
Yeah, and EPA keeps raising the tolerance levels, the amount that's in our food, because they say, well, we're getting levels that are so high that we can't sell it safely unless we increase the tolerance levels. So in May, they doubled the tolerance levels. We started out in 1974 with a tolerance in food of 1/10 of a part per million max. We're up to as much as 400 parts per million now.
So in your opinion,
Yeah, and EPA keeps raising the tolerance levels, the amount that's in our food, because they say, well, we're getting levels that are so high that we can't sell it safely unless we increase the tolerance levels. So in May, they doubled the tolerance levels. We started out in 1974 with a tolerance in food of 1/10 of a part per million max. We're up to as much as 400 parts per million now.
so in your opinion,
The companies come to EPA and say, we need to increase the amount of glyphosate permitted in our various crops. And EPA says, well, is it safe? And they say, yes, it's safe. So they keep raising it because they say, well, these are levels that we're finding in our food, and we can't sell it unless you raise the tolerance levels. EPA and FDA both have put a statement. When they approved the higher levels, they state, if I can paraphrase it close enough, that FDA and EPA does no independent testing of the safety of these levels of glyphosate in the food. We rely solely on the statement of the companies that it's safe. Now, in last May, in doubling the amount of glyphosate that's permitted in canola oil and in refined soybean oil, for instance, they apologized to the European Food Safety Committee and said, I'm sorry, but our levels have to be almost 3 times what you permit in Europe because we can't get levels that are lower than 20 parts per million. European Food Safety says 7 parts per million is maximum for them. But then the EPA also said that it's okay to have 40 parts per million of glyphosate in refined soybean oil. Well, you ask the question, why is it twice as toxic in canola oil as it is in soybean oil? And then you realize that for all of these different levels that they have all these tables for in the Federal Register of tolerance levels for glyphosate, none of them are based on science. They're based on how much we're finding in the food product, and therefore, in order to sell it, the level has to be raised.
well, in your opinion,
The companies come to EPA and say, we need to increase the amount of glyphosate permitted in our various crops. And EPA says, well, is it safe? And they say, yes, it's safe. So they keep raising it because they say, well, these are levels that we're finding in our food, and we can't sell it unless you raise the tolerance levels. EPA and FDA both have put a statement. When they approved the higher levels, they state, if I can paraphrase it close enough, that FDA and EPA does no independent testing of the safety of these levels of glyphosate in the food. We rely solely on the statement of the companies that it's safe. Now, in last May, in doubling the amount of glyphosate that's permitted in canola oil and in refined soybean oil, for instance. They apologized to the European Food Safety Committee and said, I'm sorry, but our levels have to be almost 3 times what you permit in Europe because we can't get levels that are lower than 20 parts per million. European Food Safety says 7 parts per million is maximum for them. But then the EPA also said that it's okay to have 40 parts per million of glyphosate in refined soybean oil. Well, you ask the question, why is it twice as toxic in canola oil as it is in soybean oil? And then you realize that for all of these different levels that they have all these tables for in the Federal Register of tolerance levels for glyphosate, none of them are based on science. They're based on how much we're finding in the food product, and therefore in order to sell it, the level has to be raised.
Well, in your opinion,
The problem is that our GI tract organisms are very sensitive to glyphosate. They apologize because exports are prohibited, or import into those countries is prohibited if it's above that level. We had 1 million pounds of California almonds rejected last year because of excessive glyphosate. It was above the level that the European Food Safety Authority says is safe. Therefore, when they got to Europe, somebody tested them, and they said these levels are too high, and they rejected the whole 1 million pounds. In checking the California orchards, though, the orchardist said, well, we haven't applied any glyphosate for 2 years. How did we have these high levels? Well, they have those high levels because glyphosate is a persistent chemical in the soil. It may be detoxified as an herbicide. It's not detoxified as far as it's being taken up by the plant and then accumulating in those growth points, which is in almonds is going to be the seed or the nut that is produced and marketed then from that area. So, and the other area is the orchardists are using a lot of related compounds to glyphosate as fungicides. So they use Alliette, a phosphite fungicide, that then in combination with other chemicals can essentially form glyphosate in the soil and have it taken up. So there are various sources, because we've been using glyphosate now for over 35 years, and it's been accumulating in many of our soil situations. It's not degraded rapidly. In fact, Monsanto has lost both of their lawsuits where they claim biodegradability. It's been found that's fraudulent advertising. Even though it may biologically degrade, it's an unpredictable entity, anywhere from 1.5 years to 22-year half-life, which means, oh, how about the other half? Usually the second half-life takes longer than the first half-life to degrade. So you've got 40 or 50 years that you could have this compound still present in your environment.
Okay.
I mentioned that we have over 40 plant diseases that are increased in severity since we've started using glyphosate, or that we see glyphosate predisposing to these diseases. With some of those diseases, we see some unique symptoms that 30 years ago or 40 years ago we didn't see with these diseases. Gossa's wilt on corn, for instance, used to be a very wimpy disease. Now it's a very serious disease for us. But rather than just the brown streaks and the wilting and typical Gossa's wilt type of symptoms that we had 30 years ago, we see additional symptoms now. We see a water soaking that is much more extensive, and you'll see a gray-green extension of the brown lesions, so that it looks like the leaves in the middle of July with Goss's wilt have been frozen. And then you get that water soaking and the gray-green type of color and appearance to it. And that will quite often now turn into a pink color. The whole leaf will turn pink. Those aren't typical symptoms that we used to see with Goss's wilt 30 years ago. In examining those tissues, one of the things that we see with the genetic engineering is that there's another entity that's associated with that. It appears to be a physiological entity. It can be observed if we magnify it about 40,000 times. We have been able to now grow it in a somewhat culture condition for 1 or 2 generations, but at one time we thought maybe it was a new organism associated with the Goss's wilt. And you'll see that quite often with diseases. You'll have a little different symptom when you have 2 diseases working together than when you just have one. What we find with this entity is that it can increase to very large concentrations in the tissue. When we streak it on certain media, that it can multiply, it's self-replicating, but it doesn't contain any DNA or RNA. So it's not a living entity, but it's an entity that can self-replicate in the biological system. There are 2 entities of this type that we know in nature that occur. One of them is a prion. That would be similar to wasting disease or Creutzfeldt-Jakob disease or mad cow disease, those types of diseases where you have a natural protein, in those cases in the brain. Those are metalloproteins. The protein in the brain that prions develop from is a copper metalloprotein. There are various hypotheses of what that copper does, whether it's stabilizing the protein and its function in the brain, or whether the protein is actually maintaining what we call homeostasis, or a normal level of copper so you don't get toxicity from the copper in the brain. There are various hypotheses there. What we know is that when you substitute copper for manganese, or manganese for the copper, in that area, then you get a folding of the protein, and that makes the prion. The prion then becomes resistant to most of the chemicals that would normally degrade protein, the proteolytic enzymes, temperature. You can autoclave a prion and it still self-replicates in a biological system. So that's one possibility. The other possibility is a biomatrix. Now, a biomatrix also involves protein and a metallic component, a metal component to it, or an element in that area. But with biomatrix, we see that associated with rheumatism, with arthritis, with arthrosclerosis, and also with Alzheimer's disease. So that what happens there is that you have a nano-sized crystal, metal crystal that is growing, and as it grows then it chelates the amino acids that are in a biological system, so it forms the protein. So in the one case you have a protein that is formed in normal functioning. In the other case you have essentially a protein that is being formed through growth of the crystal in a disease type situation. It gets a little more confusing where
Right.
now they're starting to refer to Alzheimer's as maybe the precursor for a prion type disease. So there's just a lot we don't know. We're still struggling with that. But what we do know with these new symptoms and the plant diseases, the much more extensive damage from these diseases, is the glyphosate appears to be associated with it, and we see it associated especially with the genetically engineered plants seem to have an altered metabolism or an altered environment that permits this type of protein development to proceed.